So good morning, everyone. Welcome to Research Grand Rounds. We have several of these during the year that really serve to highlight the ongoing research in our department. And this morning, our Grand Round speaker is Dr. Teresa Kim from the Division of Surgical Oncology. She started her career at Harvard Medical School after attending a little-known undergraduate called MIT. She then proceeded to Mass General for her surgical residency, and during her research time, did a quick postdoctoral fellowship in immunology. She then proceeded to Memorial Sloan Kettering for her fellowship in complex surgical oncology, and then stayed on there for several years as an instructor prior to joining our faculty, which was very fortunate for us, in 2017. So Dr. Kim is one of our physician scientists in the department. Not surprising, she has many current papers to her name, more than 25 peer-reviewed articles. She currently has over $200,000 of funding as a primary investigator and is also a co-investigator on almost $2 million worth of projects with others. Her two primary projects are working with immunology as well as macrophages in approaching cancer. So as we were kind of leading up to officially starting this morning, I was talking with Dr. Kim and telling her how dwarfed I so often feel when I hear some of the amazing research that's going on in our department. And so Dr. Kim, we anxiously look forward to hearing more about what you're doing.
Well, thank you, Dr. Marquardt and Dr. Wood, and all of you for logging in so early here and the opportunity to speak today. I am going to, I have no financial disclosures. As Dr. Marquardt said, I am trying to be both a cancer surgeon and a tumor immunologist and a wife and a mother to a crazy toddler here. And over the next hour, what I was hoping to do was threefold. I do want to give us each an opportunity just to reflect on purpose, because I found that it has been so important on my own path in this crazy surgeon scientist road that I'm on. And I think important to each of us as we choose our way in academic medicine. I will share many of the lessons I have learned from mentors, including many of the folks here in the audience today, and then provide just a little snapshot into the state of cancer research, the challenges, and our unique approach to studying one of them, which is immune resistance in human cancer. As an outline, again, we'll focus just a little bit on purpose, because that's really the central focus of everything. I will tell you about a topic near and dear to my heart, which is macrophages, and then a bit about how we're trying to program tumor-associated macrophages in human cancer models, specifically in colorectal liver metastases, and then some conclusions and lessons learned. If you remember nothing else, I think remember the beginning and the end. So to start with, my brother actually sent me a podcast recently that I just included down here from the Hidden Brain series about cultivating your purpose. This has nothing to do with academic medicine, but I found to be quite helpful. And basically, it speaks about how a sense of purpose is a sense that you're oriented to something bigger than yourself, and not just an achievable goal like graduating residency, but some type of lifelong goal around a question or a challenge or a vision that you really want, for example, in the case of academic medicine, to do to improve human health or how we treat patients. So when I was thinking about my own academic mission or purpose, I think it comes down to the bigger goal of alleviating human suffering, and to try to start thinking about the how. For me, what I wanted to do was improve how we treat cancer, which really comes backwards to how we study cancer, and try to do it in the smartest way possible. And so for my research interest, what I hope to do, and I'm currently doing, is using patients' tumors, so human tumor and tissue material, to identify and target the most relevant immune evasion mechanisms. The goal ultimately is to try to get to a state of precision medicine instead of a one-size-fits-all type of treatment. The interesting thing is that over the past 10 years or so, we've had incredible technological advances in biomedical research. And so this spans everything from precision gene editing with CRISPR technology, machine learning, on the bioinformatics side, and single cell RNA sequencing and multi-omics technology that really has allowed us to look at the single cell level at protein and gene expression in a way we were never able to do earlier. At the same time, on the clinical side, there's been an explosion in immune and targeted therapies, particularly for melanoma, but for other solid tumors as well, that span the immune checkpoint inhibitors like anti-CTLA-4 and anti-PD-1, which were awarded the Nobel Prize last year, as well as targeted therapies in the BRAF and MEK pathway. So despite all of these technological advances, cancer is still a major cause of both death and suffering in our country and around the world. This I don't need to belabor with this audience here, but it really begs the question, with all of these technological advances in the recent decade, why are we still not doing better at curing patients? So there are many reasons for this. The two that I want to focus on today are, number one, immune resistance in cancer, particularly with the cancer microenvironment, in which cancers have figured out not only how to propagate, but also how to hide from the immune system or shut down the immune system, and really prevent that recognition of something abnormal or foreign, which usually happens in a normal response, for example, to injury or infection. On top of that, we don't have quite the preclinical models that we need. In addition to lots of fancy technologies, we do have many types of genetically modified mouse models, which have all kinds of very intricate, carefully made genetic alterations to try to mimic human disease. But what we're really missing is human cancer-based models that can totally capture and recapitulate the complexity of human disease. So the interpatient heterogeneity, the genomic complexity, even within a single patient's cancer. And that is a major gap to get us from the technology to the actual kind of conduct of precision medicine and precision cancer care. So the key question that I had was, within this whole question of immune resistance, how do we study macrophages in human cancer, figure out what they're doing, and in fact, often what they're doing to inhibit the anti-tumor immune response and turn that around so that we can get macrophages to stimulate anti-tumor T cells? And how do we do this in a model that actually mimics what they'd be doing in the human cancer microenvironment? And so just to give you guys a little bit of background about macrophages, I think along the vein of finding what you're passionate about, macrophages are totally unique in their diversity and plasticity, meaning they can do many, many different functions as demonstrated in these panels on the right. And they're also plastic in the sense that they can shift from function to function depending on their local environmental cues. So in panel A, we see that macrophages can sometimes be the major warning alarm for the body that there's an invader or an infection. They can stimulate adaptive and innate immunity, meaning T cells in an antigen-specific way, and then the acute inflammatory response on the innate immune side. However, they also play a huge role in homeostasis, meaning shutting down an acute immune response in many different mechanisms through secreted proteins called cytokines, ligands that they express on their surface, basically ways that they communicate with the other cells in the immune system. And sometimes this homeostatic mechanism is something that gets out of control in a pathologic way, for example, in cancer. Because of the complexity of macrophages, the field of macrophage biology has kind of leaned on a simplified paradigm for these cells and along a spectrum of M1 to M2 categorization, which really comes from in vitro studies of macrophages, in which M1 macrophages are classically activated. They're stimulatory. They're the ones that incite an immune response and stimulate T cells. And on the flip side, M2 macrophages are alternatively activated, for example, in the setting of different cytokines, and are often suppressive. They inhibit T cell proliferation, and are more often the kinds that are found in cancer. And just as a brief review, tumor-associated macrophages, or TAMs, have many different functions, as I said, most of which inhibit a functional anti-tumor immune response, but can also potentiate cancer growth and metastasis. The issue is that macrophages are complicated and very context dependent. So when I was a fellow in the lab during residency, we spent a bit of time studying the immune response to this totally rare type of visceral sarcoma called gastrointestinal stromal tumor, or GIST. But it did prove to be a really useful solid tumor model that was, despite a simple genetic single driver oncogene mutation, something that was quite inflamed and had an interesting immune response, particularly around the macrophages, in which we found that in human GIST tumors, macrophages inhibited T cells, as we found in other tumor types, when those patients had been receiving and responding to imatinib. So in the setting of a dying tumor, macrophages were inhibitory. But interestingly, in untreated tumors, or those who were resistant to the imatinib, the macrophages actually stimulated T cells. So they were more along that M1 side of the spectrum, and quite unexpected based on the current literature at that time. In addition to this, we found that there was a targeted agent called PLX3397, which is now pexidartinib. It is a multi-kinase inhibitor that is essentially a targeted therapy with a few targets, including the oncogene KIT that's overactive in these tumors, as well as CSF1R, which is a really important survival receptor for macrophages. And so we found that in our transgenic mouse model, which is a good model, but not perfect, I'll get to that in a second, the combined inhibitor was even better than the standard treatment imatinib at shrinking tumors, and in fact also quite good at depleting macrophages from these mouse GIST tumors. And so this was a really interesting signal. But even in this case, the limitation of the transgenic GIST mouse model was that they don't metastasize, they don't develop secondary resistance, and so they can't really tell us how effective this drug is going to be for the situations that really matter in the clinic. And so just in terms of brief scientific lessons learned from this foray into macrophages during residency, I found that TAMs were very complicated with that total bimodal spectrum and function, stimulating versus inhibiting anti-tumor T cells, and that it was the context that really determined what their function would be, but that kind of dependence kind of showed us an opportunity that we might have to engineer macrophages. And so this is really where I grew an interest in tumor immunology and why I'm still obsessed with macrophages. And so this is, I think, a brief reminder to the residents in the audience who are thinking about or in the process of completing their research time. Pay attention to what sparks your interest, because it may be that 10 years later, you're giving a grand rounds talk about how you're still trying to solve that problem. For the remainder of the talk, what I wanted to focus on was how we are trying to program those tumor-associated macrophages in a more accurate human cancer environment, specifically in a tumor slice culture model, and then specifically looking at a cytokine IL-10 in colorectal liver metastases. When I joined here 4 years ago, I started in the UW Time Laboratory, which did not have the fancy name back then, but it stands for Tumor Immune and Microenvironment Laboratory, headed up by Venu Plarisetti, with a lot of input as well from Ray Young, two of the surgeon scientists in the group who have been phenomenal mentors. This lab has really perfected and applied a tumor slice culture model to multiple different types of human cancers. the way, and I should say that this lab has also been fueled and energized by a lot of trainees who are here today, and also alumni who've recently graduated, including who you can kind of see in the pictures here, with Kevin Sullivan and Dave So, who have not pictured here, who have both graduated, and then Kevin Labadie and Sarah Daniel, as well as a couple of the scientists in the group with Shiyun Jiang and Heidi Kennerson, who have taught me so much and have really taken this precision slice culture model and made it quite usable. So the way that these tumor slice cultures are made, often from a surgical specimen in the operating room, there are a few 6mm punch biopsy cores, which are taken in sterile fashion back to the lab. We have a high-tech vibrotome here within a sterile tissue culture hood, which with a finely vibrating blade is able to cut slices of tissue, basically like little miniature salami slices, 250 microns thick, so 1/4 of a millimeter. And these slices can then be plated in culture in standard media, but on a little membrane insert in the well so that they can live close to that air-liquid interface. And what's amazing is that these slices can stay viable, healthy for about a week in culture and maintain their three-dimensional architecture with the cancer cells, the stromal cells, with the blood vessels, et cetera, as well as the immune cells. And so it becomes an intact tumor immune microenvironment in which you can test all kinds of different chemotherapies, immune therapies, combinations, and then use a number of different assays to look at how those treatments are working. And this is incredibly valuable in tumors in which there is a complete absence of other preclinical models. And so when I started out, and I think about 3 years ago, I spoke with you all about sarcoma slice cultures because I was taking care of sarcoma patients quite heavily at that time. There was a great need in that this was one of the cancers without good preclinical models. And it sounded perfect to be able to apply this tumor slice culture model to this disease to get to some answers about immune resistance. We had some early successes, which I have shown here, the best ones, which remained intact in terms of three-dimensional architecture and microscopic histology. After about 1 week in culture. This is with 2 different types of liposarcomas pictured here. We tried this in about 14 patients over the span of a couple of years. But what was quite frustrating was that despite some of the early successes, there was not a predictability in the viability or healthiness of these tumor slices. And so some of the tumors would grow quite well, some of them would not. Some of them, even though they were totally viable, untreated sarcomas coming out of the body at the time of surgery, were completely dead even after a day in culture. And so with this type of unpredictability, that makes for a very difficult model to use. And on top of that, there is an incredible inherent heterogeneity in these sarcoma patients with over 50 different subtypes. And the rarity makes each subtype quite infrequent in the actual number of cases that we have to study and try to optimize the model. So I was faced with a decision a couple years into practice and trying to be a super successful surgeon scientist about what to do. Do I keep troubleshooting this model? Do I pivot? I think that when you're faced with a decision like that, particularly at a vulnerable point as a junior attending, trying to kind of plant your seeds and grow your vision, it's important to do 2 things. To come back to your purpose. What is it that I'm trying to do here? And then lean on your mentors for advice after you have done your internal work, figuring out what it is that you think is the best aligned with your purpose. And so for me, what I had set out to do was to try to figure out how to overcome immune resistance in cancer, in solid tumors, perhaps in sarcoma, but generally in solid tumors. And what I needed was an accurate and reliable model to start doing some of the mechanistic work. And so ultimately, I realized that if I spent another 10 years trying to troubleshoot a model, I wasn't helping anybody really in the vein of what I was trying to do, at least in this current first step. So I made a decision to kind of pivot my scientific research to macrophages in colorectal liver metastases, which might seem totally random, but based on the accuracy and reliability of a different tumor slice culture model, which was already working well in the lab. And I am grateful for what I'm showing here is a huge number of people, but who I, even though they might not know it, consider to be part of my board of directors, mentors, sponsors, just to bounce ideas off of, and especially again in these decisions that are important branch points in either the scientific development or overall career development. Does this make sense or is this crazy? And so I appreciate all of them. And it was around a couple years ago that I totally was able to focus my clinical practice as well. And I found that to be incredibly helpful to allow space and time, and especially mental energy towards getting this kind of refocused scientific project off the ground. So just for a little bit of background in colorectal liver metastases, these are the main source or cause of mortality for patients with colorectal cancer. And sometimes, uniquely, metastases are surgically resected when they're able to, and long-term survival is a little bit better in that setting, but overall survival is still extremely poor. And these cases, particularly the more common subtype of microsatellite stable colorectal cancer, do not respond to the current immune checkpoint inhibitor therapies. So there's a huge need for better treatment. There's also a need for accurate human cancer models. And in this paper that Dr. Young and his lab just published recently, what they found was that colorectal cancer liver metastases in slice culture are a really good accurate model of the human complexity of disease. So there was a particularly interesting case they had of a patient with two different metastases, the one on the top, which shrunk on FOLFIRI chemotherapy, and the one on the left, the bottom, which grew or was resistant. They were able to sample each of these tumors at the time of surgical resection and then grow them into slice culture with the sensitive tumor shown on the left and then the resistant tumor shown on the right. And what's very interesting is that in vitro, in the culture, the FOLFIRI chemotherapy had a similar effect as it did in the body. So the sensitive tumor dropped the viability of the tumor slices and the activity or the mitotic activity exemplified by Ki67 gene expression. Whereas in the resistant tumor, those slices did not drop their viability, and in fact looked like they were quite proliferative, even in the face of FOLFIRI chemotherapy in this orange line. So all of this was very in-depth evidence that these slice cultures were kind of that elusive human cancer model that we had been looking for to study the immune response as well. And to come back to that central question that I had of how are we going to program tumor-associated macrophages in a good human tumor microenvironment, in the colorectal liver metastases, now that we had a model, we had to figure out a signal. There is a major inhibitory cytokine called interleukin-10 or IL-10, which is responsible for shutting down many types of different immune responses, both in the setting of homeostasis and also acute inflammation. IL-10 is primarily secreted by macrophages and other myeloid cells, as well as a small subset of T cells called Tregs. And many different immune cells respond to IL-10 in certain contexts, including macrophages, and then all the different types of effector T cells. This is not something that you need to know in depth, but just for the context of what we're working on, the way that IL-10 works in these responder cells is that it binds to a specific receptor that then leads to a lot of downstream signal transduction, including through JAK and STAT proteins, and then ultimately changes in gene expression based on how the STAT3 transcription activators work in the nucleus. And so as a kind of bird's eye view, IL-10 is something that, because of biology, we were suspicious might be related to macrophages. And some of these retrospective data that correlate gene expression and survival have shown that high IL-10 gene expression in bulk tumor, both in this colon cancer specific subset as well as pan cancer, correlate with worse survival. So more IL-10, worse survival. This is purely correlative, but again, something that piqued our interest about this cytokine. And so we hypothesized that IL-10 is a required but reversible mechanism of immune suppression in these liver metastases, both by inhibiting the T cells directly, but also driving this inhibitory M2-like macrophage programming. This finding that I'm going to explain here is the major finding on which all of our current work is based. And this is something that Kevin Sullivan and Xiun Jiang in the lab spent literally years working on. But essentially, in a large cohort of 32 colorectal liver met patients, tumor slice cultures were generated, and they were treated with either a control antibody or a blocking antibody against IL-10 for about 1 week, after which the tumor slices were harvested and then looked at by microscopic histology, specifically cleaved caspase-3 staining, which is a marker for tumor apoptosis. And so the really exciting finding was that across these totally heterogeneous patients with all kinds of different preoperative chemotherapy regimens, et cetera, there was basically a doubling in tumor apoptosis in the slices treated with anti-IL-10 versus control. You can see each dot here represents a different patient's tumor, and these are representative images at the top showing the normal tissue histology or architecture that is preserved in the slices even after 6 days, that is totally annihilated by the anti-IL-10 treatment, where we just see necrosis here. As a second very interesting finding, anti-PD-1, which is a clinically available immune therapy that does not work in these patients, similarly did not generate increased tumor apoptosis compared with control. So this was a good reality check, again, that we were studying a signal in a model that was reflective of human disease. We were wondering, how does this happen? And we had our obvious hypothesis that this was immune mediated. So the first thing we asked was, is there more immune activation in these tumors? And in fact, we saw across both adaptive and innate immune cells that there was evidence of this. And so again, in the same experimental setup, with colorectal liver met patient slices treated with control or anti-IL-10. The anti-IL-10 slices demonstrated a greater than 2-fold increase in the frequency of CD8 positive T cells, which are a marker for the killer or cytotoxic T cells shown here, as well as increased expression of HLA-DR, which is one of the proteins within the MHC class II family, MHC standing for major histocompatibility complex, which is a really important antigen presenting molecule for the so-called professional antigen presenting cells like dendritic cells and macrophages to stimulate helper T cells. So through both of these findings, we at least saw some indirect evidence of immune activation. But what we wanted to know mechanistically was, is the immune system required for this big anti-IL-10 effect? And if so, which part of the immune system, the adaptive T cells or the innate immune system? And in this really well-done experiment with a few different blocking antibodies, which actually Kevin Labadie and Sarah Daniel helps quite a bit with as well, tumor slices were treated with either the standard control or anti-IL-10 treatment, in addition to a blocking antibody against either MHC class I needed for the killer T cell function or MHC class II needed for the macrophage helper T cell interaction. And what we found was that blockade of either class 1 or class 2, shown in the right-sided bars here, totally decreased the anti-IL-10 effect. So we saw the increased apoptosis with anti-IL-10 alone, and this, again, was totally abrogated by the dual blockade of either class 1 or class 2. And so this was truly the piece of mechanistic information that we had, that when we take away that IL-10 signaling with IL-10 blockade, it requires this concerted standard immune response of both killer T cells, helper T cells, and antigen presenting cells coming together to induce this anti-tumor mediated killing of the tumor. And so the next question, or next set of questions that I'm currently working on, is figuring out a little bit more into the mechanism on the myeloid or macrophage side. How is this working? And backwards, what is IL-10 doing at baseline to potentially kind of perpetuate its own expression in the macrophages? This is what we think is going on in cartoon fashion, that basically this giant purple cell here is a schematic of a macrophage. And we think that these are the major source of IL-10 in the tumor, or a major source at least, and that their own either autocrine or paracrine signaling of IL-10 is leading to downstream JAK-STAT effects in gene expression that are ultimately driving more IL-10 expression, as well as additional suppressive features. And so there's two main arms of the project that I'm currently working on as kind of the first step in trying to understand what is going on with the IL-10 in the macrophages. Why are they making it? What exactly is that signaling doing inside the macrophage? And I will say, as an aside, an important lesson that I think that people like Venu and Ray and other mentors still have to remind me is, once you find your purpose and your focus, try to stay focused and answer the first question in line. A lot of times it's very easy to start jumping 3 steps ahead to thinking, well, how will I translate this and make this an awesome cure for cancer patients? But really, there's probably a first question that you need to figure out as your aim 1, aim 2, et cetera, to kind of prove your assumptions that you're making. And so for us, for these colorectal liver met macrophages, what I needed to figure out first was which macrophages were in the tumor, what did they look like, what was their function, which ones were making the IL-10, ultimately, why is that, to get towards other potential therapeutic targets. And so what we're seeking to do is take a cohort of colorectal liver met patients, kind of prospectively get their tumors, process them into single cell suspensions for really high dimensional single cell RNA sequencing profiling, which in the biomedical research world is the hot new technique now. But it is quite powerful because you get gene expression in thousands of different cells that you can then kind of glean what populations are present in a much more specific and granular way than before. And to look at spatial analyses of where these IL-10 positive macrophages are within the tumor. The separate question that we're working on in parallel is, what is IL-10 doing in the macrophage exactly? And when we block the IL-10, how does the gene expression and protein expression / function of these macrophages change? And that's where the tumor slice culture model is going to be extremely important. And so, again, as a brief aside, when setting out to start new aims of this project, I realized that there were things that I knew well from training, like basic macrophage biology and lab techniques. There were things that I was starting to learn in the tumor immune microenvironment lab here with the Time Lab team and the slice culture technique. And then there were things that I had no idea about, including high dimensional genomic analyses and everything in computational biology. And so I have found over the past couple of years that one of the incredible things about being here at the Hutch at UW Cancer Consortium is that there are people across the consortium and even beyond in some of our Seattle collaborator institutions that have deep expertise in these areas, from whom I have been able to learn quite a bit. And in the case of this project, where we've been able to collaborate and get some preliminary data to work with. And so towards that first aim of just figuring out which macrophages make IL-10, we set out on a pilot experiment where we took 4 patients' tumors, colorectal liver metastases, digested them into a single cell suspension, and then ran them through a platform in which we could get basic information about 50 different surface proteins and 400 different genes, again, on a single cell level. And what we were trying to figure out was, if we looked at the myeloid cells, which were primarily macrophages in these tumors, could we essentially figure out what genes were differentially expressed between IL-10 positive and IL-10 negative macrophages, just to get to the place where we knew what kind of phenotypic markers we could use to pull out these IL-10 positive TAMs. And so these plots here look similar to flow cytometry plots. Each dot is a cell in this pooled suspension of tumor cells from these patients, with each axis showing a particular protein marker. CD11b is a major myeloid marker. And within this population, shown blown up here, we could look at IL-10 expression on the x-axis. And it does seem like there's a separation of populations between IL-10 positive and IL-10 negative macrophages. But when we looked at the differential gene expression, which is shown here in this volcano plot, what I was expecting to see is that here on the right side showing fold change increased genes, and then here on the left decreased, I was expecting to see many blue dots, meaning lots of differentially expressed genes, either up or down. And unexpectedly, what we found was almost no differentially expressed genes, just a few chemokines related to neutrophil in infiltration, but really nothing else except for IL-10, obviously, because that was how we defined these populations. And so this was an interesting finding that totally disproved my hypothesis that there were going to be different populations of macrophages secreting or not secreting these IL-10, this IL-10 cytokine. We tried to do slightly more complex analysis with unsupervised clustering of the macrophages shown here in this UMAP plot. But interestingly, instead of seeing like, oh, it's just in the blue cluster here that we see IL-10 shown in red, the IL-10 was distributed across all the different clusters, which is also shown here on the right graph. And we really saw expression in clusters related to totally different kind of functional enrichment in terms of macrophages related to acute inflammation, T cell stimulation or antigen presentation, or the more potentially M2-like inhibitory macrophages related to tissue repair and remodeling. So at this point, there are questions that remain about how I suspect that for the IL-10 expression in these tumor macrophages, it's a dynamic function that really depends on local microenvironmental cues at any given place and space and time. And so what we need to do at this point is look at a larger cohort of patients' tumors through this same single-cell RNA sequencing platform to see if we can validate this finding, and then look towards our spatial analysis to see where these macrophages are, because that may give us some extra hint as to the cause of the IL-10 expression. Are these macrophages with IL-10 more closely associated with apoptosing carcinoma cells? Is this something related to phagocytosis of dying cells, which is a running hypothesis that we have now as kind of a new version of a hypothesis? And then to look at clinical variables related to what the patient and the tumor looked like before surgery. Did they get a specific kind of chemotherapy was it that there was a major response to the chemotherapy, and that's why the macrophages are doing what they're doing in tumor slice culture. And so this is kind of what's next for this aim. And as a brief preview of the other side of what we're trying to do studying these macrophages, in the tumor slice culture model, to figure out more specifically what the mechanisms are of IL-10 signaling or IL-10 receptor signaling or blockade, we're setting up prospective tumor slice culture experiments with a few different reagents to try to tackle different steps in the IL-10 receptor axis. Meaning, is it that we get the most change in phenotype and function of the macrophages, taking out the ligand, which is primarily what we did in preliminary data, or taking out one or both of the receptor subunits, or doing something downstream blocking JAK1 or STAT3. It is, I think, going to be of key experimental importance and potentially therapeutic importance to be able to target this IL-10 axis in a cell type specific way, to know, is it that inhibiting the IL-10 receptor in the macrophages is most important, or is it that we have to inhibit it in both the T cells and the macrophages? Is very difficult to figure out how to do this in a tumor slice culture model, because it's not like a mouse where you can just knock out a gene or make a transgenic model. These are human cancers that come with their own complexities all bundled in. But we have been able to make an interesting new collaboration with a protein design group at Stanford, who recently published a paper in which they're starting to be able to develop different IL-10 receptor binders that mimic the IL-10 structure, but slightly tweak it so that they can have different binding affinities and function downstream. So I think that we're one step closer to potentially trying to target myeloid IL-10 receptor signaling. And we have, I think, our first experiment cooking in the histology lab right now, trying to figure out what the effects were. So this is also a preview of coming attractions. And I think, as I said, we're one step closer to figuring out, at least in colorectal liver metastases, how to target IL-10 to program tumor macrophages to be better T cell stimulators. And I think if we're able to do this, then there's a whole next set of aims that we need to figure out related to how this interacts with standard chemotherapy, what the best reagent is to test in longer term models, for example, mouse models, before trying to make the jump to clinical trial, which would ideally be in a neoadjuvant window of opportunity type setting, so that we could study the tissue before and after as well. And just in terms of my own timeline, this is mostly to show the trainees that in addition to trying to figure out your purpose, once you do that, that is what should kind of direct you to get organized, and then to try to set a timeline of milestones. It's quite empty to set the timeline of milestones if you don't have a sense of your purpose, but you kind of need both. And so over the next several quarters, I'm going to be trying to analyze some of these data. And I'm totally grateful for the support so far from the Department of Surgery, as well as the Cancer Consortium, to be able to do a lot of this work. And these are just many of the grants that I'm trying to apply for. I think Dr. Flum has told me repeatedly, you can't get a hit if you don't go up to bat. So I've been up to bat many, many times and failed many times. And it's quite fortuitous to have, I think, a team and mentors to remind me that that's normal. Then you just have to keep going and improve your strategy at each iteration. And so just to wrap things up here and leave a few minutes for questions at the end, there is a set of scientific lessons that I've learned, and then a set of career lessons. And so just in terms of the science, to remind you all, over the past hour, I've told you how macrophages are totally messy. Cancer is messy, human disease is messy, and macrophages are very messy. But their functional diversity and plasticity is really what makes them a potentially very powerful tool in cancer immunotherapy, and worth getting through the mess to figure out how to utilize them. Studying the TAMs in human cancer models is really essential, as I learned from early experiments in the GIST mouse versus patient samples, and now what we're seeing here in colorectal liver metastases. And my ultimate vision, once I figure this out in this particular disease context, is just more generally for solid cancer patients who don't have good systemic therapies at advanced stage, how to make a really effective combination of high dimensional tumor profiling of patients' tumors with functional interrogation of these targets in a tumor microenvironment that's accurately reflecting human disease, for example, a tumor slice culture model. It's really the combination of that that's probably going to tell us what therapeutic targets we can really focus on to have a higher yield of success in clinical trial eventually. This all ultimately is hopefully to get us to the point of trying to alleviate suffering in patients with these advanced stages of cancer. This is just an example of how that program, which doesn't exist anywhere except in my head, is going to play out eventually. And in terms of career lessons learned, these lessons are, as I said, an amalgamation of many different mentors that help along the way, as well as speakers in this forum who have shared their own experiences. I think a lot of us remember Dr. Bulger's talk about the why when she was talking about her, I think, incredible work in trauma. So no matter what you call it, purpose, meaning, calling, the why, I think the most important thing is to remember, why are you here? Why did you choose academic medicine and potentially a big pay cut to do something or tackle a challenge to improve the human condition? Once you figure that out, everything follows in step. So number two, if you have a sense of your purpose, you can map out your path. What are the milestones you need? What's the timeline? And importantly, when new decisions or problems come up, how do you actually tackle that decision based on your purpose and try to maintain your focus? Number three, which is totally key, is to build the team. So supplement the scientific skills that you need, kind of build your board of directors so that people who care about you can give you advice when you're trying to navigate waters that are new and foreign, and surround yourself with what I signed up for this academic coaching program, and she calls these people informed believers. Find people who understand what it is that you're trying to do and to whom you don't need to prove that you can do it so that you can get to the strategizing. This is, for example, not just your parents who think you're great. And then importantly, as you're doing all of this, take some time every week or every quarter, every year, whatever it is, to reflect and then iterate on steps 1 through 3. And definitely recognize those who, for whatever type of altruism, decide to help you along the way. And this is just a brief acknowledgement of many of those people, including the Time Lab and Dr. Pilarsetti and Dr. Young for their kind of incredible mentorship along the way. The residents in particular with Dave, Kevin, Sarah, and others who are embarking on joining the lab for all their hard work and their curiosity, which is really, I think, one of the driving things for a lot of us in science. And then many of our collaborators, current, past, in this institution and abroad, and as I said, of funding from the department, which has been key in starting this whole process, as well as some funding from the Cancer Consortium. And I think more broadly also, thanks to the Department of Surgery community, especially our GSD division, for being a work family away from home, the residents, and my own family for that other half of the purpose here. This was the picture that my son picked. And with that, I am happy to close and take any questions.
Dr. Kim, thank you for a fantastic presentation. Not only did you educate well and make this understandable for us who do not foray into basic science every day, but I loved your sort of career lessons. I'm going to open the floor for anyone who has a question they would like to ask themselves, and then I'll continue to monitor chat and present other questions. If no one else is going to go first, I know you kind of touched on this in the end, but I thought your lesson of kind of pausing to maintain focus is so critical, no matter what kind of research you're doing. Whether you're doing basic science, outcomes, education, et cetera. Kind of how do you actually do that stop to focus to make sure you're not leaping ahead 3 questions, which is so easy to do? Is that a personal focus for you? Is it a combination of that and mentors? Sort of do you have kind of an example of how you've done that to kind of maybe provide a lesson for other faculty and residents?
It's a very good question that I continue to struggle with. But I think in a particular project, what's been helpful is grant writing, actually, where I think I had gotten advice a long time ago that you can use grants. They're not a goal so much as a stepping stone that you need to be able to do the work that you want to do. And grant writing is a tool to plan your projects and experiments as well. And so I think getting some specific aims down on paper and vetting them myself, and then going through a second round of feedback with scientific mentors who understand the field, or those who don't, has been helpful in that first gut check. Am I focusing on the right thing, or am I 3 steps ahead of myself? And so getting the thoughts and aims down on paper is surprisingly difficult sometimes, because you have to commit to something and put it out there. This is something that Dr. Polarisetti has been particularly helpful in doing, but for a particular project, that's been helpful to maintain that focus. For the overall long-term game, I think that for me, having quarterly check-ins with myself and with my mentors in different capacities has been helpful. But that is on the assumption that at the beginning of the year, or essentially the end of the prior year, I've spent some time to be like, here's what went well this past year, and here's what I want to do over the next year to make sure I'm getting to the point of where I want to be or what I want to be doing in 5 years. And so I found it helpful at the beginning of the year, whatever it is, the calendar or the academic year, to say, these are my goals. And if that's the case, these are the things that I probably need to do at these 4 checkpoints of the quarters. And then trying to force myself to have a little bit of internal CEO time for an hour at the beginning of a quarter to make sure that I'm vaguely on track. So in theory, that's a system that is working for me. I think for others, you have to figure out how to stick an hour on your calendar with the interval that you want to make sure that you're on track with your goals.
I think Dr. Bird has a question, and then I have one from chat.
Good morning, everybody. Teresa, excellent presentation, clear, articulate, flowed logically. Nice job. My question is, when you hit the point about 3/4 of the way into your talk, when you said this experiment completely disproved my hypothesis, I want to know how that hit you. Was this how could my hypothesis be this far off? Was it well, this is interesting, or was this an opportunity to go a different direction? How did that hit you from a gut perspective and a strategic going forward perspective? Thank you for your talk.
Thanks, David. Thanks for joining on vacation also. I initially, when we got these results, I want to say that I thought, oh, this is so fascinating. I think my gut instinct initially was, oh no, like, what am I doing and how am I going to get these macrophages isolated, because I can't even figure out what the markers are. And so I was a bit taken aback / unhappy. But I think in taking a step back and figuring out what to do next, it was in the second iteration of looking at data that I found this to be interesting, to open up a pathway into figuring out the truth, which is basically what all scientists are trying to do, is figure out what the true story is and not what the made up one is. And so I think that looking over data with people of different perspectives was helpful to get towards that current state of mind.
We have a question from Dr. Mogul. It says, have you considered potential vehicles for anti-IL-10 inhibitors? Would you consider perhaps down the road that the therapy will likely be regional or systemic?
Yeah, it's a really good question, Harvesh. I know you're always thinking about regional treatment options. With IL-10 functioning in such a homeostatic role, there are probably potential systemic toxicities like autoimmune reactions, similar to other checkpoint immune therapies that would happen if we tried to give this blocking antibody systemically. So regional delivery to the liver would get rid of some of the systemic toxicity problems, but probably still predispose people to really bad autoimmune hepatitis, for example, through hepatic arterial infusion pumps that some folks are working with. And so I think the ultimate goal is to try to get to a point where we can have a site and tumor specific delivery of IL-10 blockade. That's kind of like a big goal that some people like Chris Garcia's group may actually be able to help us with.
There's a question from Dr. Wu. It says, what are the limitations of tumor slice technology to test individual patient tumor responses to available treatment options?
It's a really good question. Thank you, Peter, for I think that ultimate goal of precision oncology. There is a need right now for enough tissue to grow these slices in a way that still gives a window to much of the tumor. So surgical specimens have been the most usable tissue source. There many times has been a question, can you develop some kind of similar model from a little core needle biopsy? But it's such a small amount of tissue that I don't think anyone in the lab has been able to kind of reproduce the same information from that smaller piece. And so this would be limited to people having surgery for their cancer treatments, which is not a lot of metastatic patients. So I think that's one limitation. And the other is that there's not yet a good way to cryopreserve these tissue slices and bring them back to life, so to speak, to try a new treatment a few years later if there's a new treatment available or a new clinical scenario for that patient. So some of those things I think are kind of maybe a next step in this human tumor modeling.
So I'm going to combine to some degree the next two questions. There's one from Scott Breckenridge. Welcome back, by the way. And as well from Dr. Olschlager. From Dr. Breckenridge, he says, you know, as a newly arrived or say returned investigator, perhaps tips on plugging into collaborations at UW outside of your methodological area. And I think it's really a good follow-on from Dr. Olschlager to that, to say, how do you sort of, what's your advice for surgeons trying to mesh science and clinical practice?
Oh yeah, these are huge and great questions that I wish I had more time to tackle. Within a big institution / consortium like this, it has taken years to figure out where these people are hiding sometimes in terms of potential collaborators outside of the initial area. In the cancer world, the STTR, the Seattle Translational Tumor Research Network through the Hutch has been very helpful. And I think some of the Cancer Consortium and other weekly lab meetings or seminars that are kind of given through some of the different departments. And so it often has been talking to somebody or hearing somebody give a talk and then getting in touch with the other person to finally the person who is helpful. And I would say, as a side piece of advice, be willing to have a few conversations to find the right collaborator. They're there. And the beautiful thing about this community is that they're usually excited to collaborate. It's just that it's sometimes a 5-step process to find them. I think there's probably analogous kind of consortia in the topic that you may be of interested in that's not necessarily cancer related. The BBI is the other one that I was thinking about. The Brotman Beatty Institute is an intramural funding kind of collaborative institute that's trying to bring people together along the lines of precision medicine. And so a lot of the computational strength of the institution is interfacing with there, and another way to meet people. And I think with Dr. Olschlager's question about trying to mesh the science in a clinical practice, like I take care of skin cancer patients and I'm studying macrophages in colorectal cancer liver metastases, which again, in some ways seems totally disconcerted. But I think if you are somehow able to glean from your clinical practice the questions that matter, if that is your strength as a clinician scientist, and then figure out on the science side how you want to try to answer those questions, it may be that you have to take a slightly circuitous route to get to the point of studying the exact patients that you're taking care of. But for example, in the cancer world, solid tumors have quite a number of parallels across different diseases. And so that is kind of my current approach right now. And it may be that I get to study macrophages in checkpoint resistant melanoma patients a few years down the road.
So I think we're just about out of time, but thank you so much, Dr. Kim, for a truly fantastic presentation this morning, really enlightening us both on the sort of how to do it as a job in addition to the actual science. I do want to remind everybody, please do check your calendars for the resident review link that actually is starting now. Great to see everyone. See you next month.
Thank you, guys.